Hi everyone. In the last video, we saw how the secondary immune response is much greater and faster than the primary response. Now we will see how this applies to the practice of vaccination. This is what the exam questions tend to focus on for this concept. The aim of vaccination is to stimulate a primary immune response without causing disease. To achieve this, an antigen is introduced to the body as part of a vaccine. This stimulates the primary immune response, producing antibodies and memory cells, as we've seen in earlier videos. On subsequent exposure to the antigen, for example as a result of infection, these memory cells rapidly divide to produce plasma cells, which in turn secrete antibodies both quickly and in high concentration. This enables the pathogen to be destroyed before it has a chance to replicate and cause the symptoms of disease, and the individual is unaware they've even been infected. Vaccination also provides protection to a population. For a person to be infected, the pathogen must be transmitted from an infected person. In a population where individuals have no immunity, that is, no memory cells to the specific antigen, the infection can spread quickly, causing disease. Each infected individual must undergo the primary immune response, which, as we've seen, is slow before the pathogen can be destroyed, and this allows more time for the pathogen to be transmitted to another person. If a significant proportion of the population is immune, either by having already recovered after natural infection or by vaccination, any encounter of the antigen stimulates the fast secondary immune response, so it reduces the chance of the pathogen being transmitted, with immune individuals essentially providing a barrier between susceptible and infected individuals. This concept is known as herd immunity. The proportion of the population that must be immunized in order to reach herd immunity varies for different pathogens, depending on the ease and mode of transmission. In the next video, we'll look at one reason why developing vaccines can be difficult with the concept of antigenic variability.